Welcome to the Kendall Arthur Smith video conferences. This week, we're going to be talking about COVID again. You know, I don't think it's ever going to go away, right? And given the fact that we're right in the midst of a huge upswing in the number of new cases due to the Omicron variant, and because of the fact that it looks like I waited a little while, but it looks like the number of hospitalizations and deaths are not just following in sync as they have in the past outbreaks and so forth. And so then the question is, why is that? The other thing that's happening with Omicron that's new is that the vaccinated people that got vaccinated over the past year are getting infected, and that was down to a very low percentage of total patients or new infections were really from vaccinated people. Most of the new infections since the introduction of the vaccines have been in non-vaccinated people. So then the question is, why is that? And then why aren't they getting really sick? And so that all relates to T cells. And T cells are named that way because they are the lymphocytes, the precursors of T cells, arise in the bone marrow like all the rest of the blood forming cells, and then they migrate to the thymus. And nobody's ever explained to me how they know where to go, but they end up in the thymus, and in the thymus they get educated to be T cells. And so they matriculate into it's sort of like a university for T cells, and they spend a fair amount of time there. And they undergo a maturation process that takes a while, and they migrate, trickle in and out of the thymus. The thymus is a collection of lymphocytes in the upper chest. It sits right on top of the great vessels as they come out of the heart. For a long time, people didn't know what this thing was for, or why it was there in the first place. And they used to irradiate the thymus. Little kids back in the early days when they had irradiation, and they were irradiating all kinds of things because they didn't know what it was for, essentially. So 2 years after the pandemic has been with us, we finally have in the news now, and that's how I gauge how things are going. They've started to talk about T cells. And so since I've worked on T cells my entire career, I finally said, maybe there is a God. It's finally, the rest of the people have discovered T cells. One thing about the infection of COVID that's very, very important to understand is that the symptoms that bring you to the doctor and to the hospital and to the ICU and then cause your death are not due to the virus. The virus itself can replicate in your body, and does, in the so-called asymptomatic phase. So you become infected and you're asymptomatic for anywhere from a week to 10 days to 2 weeks. And then if you're going to, then the symptoms start. So why is that? And the answer is that the virus is not causing any problems. The virus doesn't have a toxin. Itself is not toxic. It doesn't cause any problems, but it's a foreign being in your body that is getting increasing numbers of players. And so what happens is the virus is very much like smallpox and measles viruses. The virus has genes that code for molecules that block our early so-called innate immune response. That's primarily interferon. Interferon is a cytokine, which means it's a small protein hormone-like molecule that was discovered in 1957 by investigators in Switzerland, when they found that if they infected a cell culture with virus and then waited a while and then took the culture medium, the supernatants that was feeding the cells, took that off, got rid of the cells, centrifuged them out, and then you use that medium, that culture broth, that's what it is. It's just glucose and nutrients and minerals so that the cells can grow. They then put that on fresh cells that had not been infected and wait a while and then infect those cells. They're protected. Something interfered with the virus-induced infection, and in this instance, they used viruses that would actually kill the cells. So they didn't know what that was. It's taken a long time to figure it out, but that's interferon. And interferon is then produced by virus-infected cells. And so in COVID, what happens is you become exposed through your respiratory tract because it's in the air. Same way with smallpox, same way with measles. It's in the air, you breathe it in, you suck it in, and it goes to all the little places in your lungs, and then is distributed throughout your body, essentially, and starts to replicate. And it just turns out that the COVID virus has a spike protein on the surface that binds to a receptor that's present on a lot of different cells in the body, particularly the blood vessel cells, the lining of the blood vessels, and the heart, the intestine, and the kidney. And so this virus sets itself up in these cells, and as soon as it binds to the receptor, which allows it to get internalized into the cell, and as soon as it gets into the cell, it uncoats and it releases its genes, and they start replicating. They start duplicating the genes, and they start transcribing or translating their RNA into protein, and they start making these proteins, and the proteins block interferon production. What could be better from the standpoint of the virus? So then there's no interferon early on in the immune response, and the virus just replicates up the gazoo all over the place for a week, maybe even longer. And then what happens is finally the more sophisticated part of the immune response finally marshals its forces. And the more sophisticated is called the adaptive immune response, as opposed to the innate that's always there. Adaptive means is that it changes, it adapts when it gets infected. And the adaptation is that it gets bigger and stronger. But that all takes the week to 10 days while the virus is replicating. And what's going on during that time is the adaptive immune response, which consists of lymphocytes, B lymphocytes that make antibodies, and T lymphocytes that have been educated in the thymus. And there's a few members of the lymphocyte genre that recognize this virus. They become activated. They start to make molecules, cytokines particularly, and these molecules start to cause the symptoms. The symptoms are inflammation, and when inflammation occurs in the lung, that's really bad because of the fact that it causes the capillaries in the lungs to leak all of their plasma and their fluid from the blood into the air spaces. And essentially your whole lungs fill up with inflammatory fluids, and you die, you drown essentially. So that's the scenario. And key in that whole thing from the standpoint of the COVID-2 is that the ability to block interferon production early on. And that's probably what the T cell immune response that I'm going to talk about is circumventing. So because we didn't really understand, most of the things that are being talked about in the lay press and journals and so forth is really what happened in immunology throughout the first half of the 20th century. Things were being injected in experimental animals or humans were being infected. For example, the 1918 flu epidemic that killed 50 million people worldwide. And there was only 2 billion people in the world at that time. So that was a huge amount of they were stacking up the bodies at the corner of the streets in Philadelphia and Boston and so forth. And people were just dying in their beds and being discovered weeks later, essentially. It wasn't good. That set up then for a generation of scientists who started to study influenza virus. And one of the major things that they were able to do subsequently, not during the pandemic, but they developed assays for the antibodies that were appearing in the blood of patients when they were infected, specifically for the virus. And so for the first half of the 20th century, antibodies was synonymous with immunity. T cells didn't even know about them. They finally had some data on T cells. They didn't know it were T cells. In 1944, during World War II, there was a young investigator in England by the name of Peter Medawar, and he was exempted from the draft, or he didn't go into the army, but he started researching things that were very important for the armed services. And what he was set to work on were methods and ways to try to use skin grafts for the soldiers that were being burned up the kazoo during the war. And he found essentially that, and I guess it was already known, that if you took a layer of skin from one individual and put it on a burn of a second individual, that the graft would look okay for about several days, maybe a week, but then it started to fall apart, and it was essentially would be crust over and be sloughed. So that was skin graft rejection, and the question is, why was that? What Peter Medawar showed in his first paper in 1944 was that and he did experiments on rabbits, if you did the same kinds of thing, you took skin from one rabbit and put it onto another rabbit, and then it would take for a while, and then it would be sloughed and rejected. Then that initial rabbit was so-called primed. If you took another skin graft from the same donor and put it on that recipient rabbit, it was rejected much faster. That was called second set skin graft rejection, and that was deemed an immunological phenomenon. And the most significant thing he showed in his paper, which was published in the Journal of Anatomy, by the way, was that he couldn't find any evidence for antibodies to be involved in this graft rejection phenomenon. And so that was strange. But that then was followed up by other investigators in the 50s, and which showed that there were lymphocytes involved in these kinds of reactions, these skin graft rejection reactions, that look, for all intents and purposes, like the lymphocytes that should be making antibodies, but they weren't making antibodies. So that came to be called cell-mediated immunity, as opposed to what was called humoral immunity, because it was in the humors of the body, the liquid portion of the body. That's where the antibodies were found. And it wasn't until the 60s and then into the 70s that things started becoming be worked out. And throughout the decade of the 60s, there was a young investigator from Australia by the name of Jacques Miller, who had moved to England for his postdoc. And he started wondering about what people had wondered about the thymus, and they'd done experiments about the thymus because they couldn't find any antibodies to speak of in the thymus either. And they thought, well, it looks like they're the same kinds of cells that we find in the lymph nodes, but there's no antibody. And they had done thymectomies. They had taken thymuses out of adult mice and then immunized them with antigens and so forth, and the immune responses seemed just fine, no problem. Well, Miller was intrigued by the fact that he reasoned out that perhaps what was going on is that the thymus, well, the other thing about the thymus that was really interesting and important is that when the animal or the human grew up and went through puberty, the thymus involuted, sort of got really shrunken down to be just a rudimentary fragment of its former self. That was strange. Because during childhood, the thymus would get very big. And that's one of the reasons that people started irradiating it, because they said, they'd take x-rays of the chest for some, for maybe pneumonia or something. They'd find this great big thymus in there, and a little, and a 6-year-old. And so they were irradiating it. not such a good thing to do, actually, because what the thymus is doing is that, as I said, it's educating the T cells. And it wasn't in 1960. So Jacques Miller published a string of papers in the 60s throughout the whole decade, really pinning this down. That if he did what he did that nobody else had done before is that he took the thymus out immediately after birth, within 3 days of birth. He devised a special method with to go in the neck, superior neck, and go in with a pipette and suck out the thymus out of these little tiny little mice. They're only as big as your little fingernail, essentially, at that stage when they were newborns. And then what happened is that those mice seemed to be okay for the first month of their lives. They grew up. They had normal growth curves and so forth, and they seemed to be all right. But then he noticed, he did a whole bunch of tests. He found that they were immunodeficient, and what they lacked was the cell-mediated immunity. They could still make antibodies, but not as well as the normal mice or the control mice that he had experimented with. In 1965, Max Cooper came out with papers in the chicken. It's interesting how we use our different species in order to find out what's going on. But the chicken, it turns out, had an organ. There was an outpouching of the colon that had a lot of lymphocytes in it. And they had actually, before Max got involved, the investigators at Ohio State had accidentally discovered that if you took the bursa out of the chicken, then you didn't have any antibody formation. So in the chicken, so what Max did was he bursectomized chickens, and then he thymectomized chickens, and he showed that there are at least two different systems going on of lymphocytes. They were the cells coming from the bursa. And those came to be called B cells because of the bursa. Then it's another story. Ultimately, they also come from the bone marrow, so that worked out for the standpoint of their name. And then the thymus was the source of the thymic-dependent lymphocytes, which became shortened to T cells. In the 60s, they kept on trying to, when you looked on a microscope, the cells looked exactly alike, the small round lymphocytes and so forth. Then in 1969 and 70, investigators, Marty Raff in England and Ben Pernis at Columbia in New York, found that the B cells had antibodies on their cell surface immunoglobulin. That then later became recognized as the receptor for antigen on each B cell. The T cells didn't have any surface immunoglobulin. And so they didn't have a marker for T cells until these people did their experiments. And they found an antigen or a molecule that was called theta on T cells. Now they had markers. They could use antibodies for B cells and antibodies to theta for T cells. And that made things a lot more amenable to further experimentations. In the 70s, after that, things really started to open up, and a series of experiments were done by investigators that showed that the cells, the B cells and the T cells, in order to have a normal immune response, they had to be from the same individual. They had to be histocompatible. If you tried to mix B cells from one individual with T cells from another individual, it just wouldn't work right. And so that was then traced genetically to the major histocompatibility locus in the genes, and that coded for molecules that were called MHC encoded molecules, or in the human it's HLA antigens. Now that becomes important because ultimately people realized that they couldn't get an immune response unless they had histocompatible cells going in their experiments. And that turns out that then in the early 80s, Ellis Reinhardt's particularly, there was a bunch of investigators who were trying to find the T cell antigen receptor. And a lot of theories at the time said, well, maybe it's a new immunoglobulin that we haven't discovered yet. Well, that wasn't true, but it was very similar to immunoglobulin. It became the T cell receptor. And Ellis Reinhardt's was the first to show in the human that the reason that you had this major histocompatibility complex story going on is that the only way T cells recognized antigen was that the antigen had to be on top of on top of an MHC encoded molecule on the surface of a so-called antigen presenting cell, of which there were the major ones were two, were macrophages on the one hand and B cells on the other hand. Then in the late 70s, 1987, there was a landmark paper published, a couple of them in Nature, from a young woman by the name of Pam Bjorkman, who was working with Don Wiley, who was a structural biologist, and Jack Strominger, who from Harvard, who was an immunologist. And they were all interested in trying to understand the structure of the MHC encoded molecules. And they came out with these landmark papers showing that it looked like a hot dog bun. And in the middle of the hot dog bun, in place of the hot dog, was the antigenic peptide. So now they started talking about peptide MHC complexes. And there was another series of investigators who realized that what was happening is that if you had an infected macrophage or B cell, or if the macrophage or B cell took in fragments of a microbe, they would chop it up into little pieces, 6 or 8 amino acids in a stretch, and that would bind to the hot dog bun. And so that's how the whole system worked out and worked together, and that there was a process particularly in macrophages and B cells that was called antigen processing, and that was chopping up of the larger proteins into peptide fragments, and the peptide fragments could be anywhere from 6 to 10 amino acids, depending on the situation. Now, going back to COVID, the spike protein is what everybody made their vaccines out of. The virus itself, that's just one gene product from the virus, but the virus has like either 26 or 29, I can't remember how many, but it's got a lot of genes coding for a lot of other proteins. But because everybody's in such a rush to get a vaccine that would protect people at least from something or other, they knew, because of other experiments done with other coronaviruses, that the spike protein was the one that allowed the cell to get inside and infect other cells. And so if you could block that, you could block infection. That would be good. And that's what happened with the initial vaccines all over the world, and all of the vaccines. And the ones that came out first were all spike protein specific. They generated what is called neutralizing antibodies. And neutralizing antibodies are called that because of the fact that the way they are tested for the antibodies is to have a cell culture and put antibodies into the milieu, and then add virus. If the virus was blocked from infecting the cells, that was a neutralization of the virus. So that's the way immunology has been since 1900. And I've sort of thought to myself, well, since I've studied T cells my whole life, what about the T cells? Why aren't we talking about the T cells? And there have been papers that have been coming out, and I've been reading them and putting them in my file and so forth. And T cells, it turns out that T cells make things just like B cells do. They don't make antibodies, but they make cytokines. And that's how how one of the ways that T cells affect their immune response. In addition, it was discovered in 1968, just before B cells and T cells were marked, that T cells could kill other cells. And there was an assay that was devised called the Chromium 51 release assay and so forth. People started studying this whole phenomenon. And to make a long story short, it took them 16 years until 1984, really, to really pin it down biochemically and molecularly to find out how these T cells could kill other cells. And they knew around 1980, because they'd done electron microscopy experiments, that the T cells that were killer cells had funny granules in them. And they looked at those granules and they said, well, I bet you those granules are what the T cell has that's able to kill other cells. What they found was that was true. And they used our original T cell lines, the CTLL1 or 2, actually, and they grew up 1 billion of these things. I just reviewed that for the Journal Club a week or two ago. They isolated these granules, and they could then show that they could, with the purified granules and adding them to red cells, that they could lyse the red cells. They could also lyse tumor cells and so forth. So that's where the money was. And they went on from there. And 1984 was when they isolated the granules. They went on from there and then isolated the proteins from the granules. And what they showed was that these proteins would polymerize into long pipes. And the pipe would be inserted into the membrane of the target cell, and it would create a hole, a visible hole. You had to have an electron microscope, but you could see that hole. And then the killer T cell would inject enzymes into the target cell, which were digestive enzymes that would digest that cell from the inside out. So that made a lot of sense. It made sense to Peter Medawar experiments from 1944. So that was 40 years, 44 to 84. That's not bad, I guess, all things considered. And so that was where T cells. Now, what I forgot to say was back in the mid 60s, in 1965, T cells were discovered if you would activate them with mitogenic proteins, glycoproteins, lectins they were called, like phytohemagglutinin. You could make T cells make interferon-like molecules. That's very important when we get back to COVID, because when we've immunized all these people with a spike protein, they've made neutralizing antibodies, but now Omicron has mutated around that, circumvented that. So those antibodies don't stick to Omicron like they do to the other ones, the earlier variants. And so the host is like a naive host again. The virus has its genes that are inhibiting the virus-infected cells from making interferon. But the difference between the immunized host and a naive host is that all of the amino acids and so forth from the spike protein were taken up by antigen-presenting cells, processed and activated T cells. So now we've got CTLs, cytotoxic T cells, and we've also got so-called helper T cells that can make copious amounts of interferon. And the interferon they make is now called interferon gamma, and that may be a little bit different kind of interferon from what the virus infected cells made. But the thing is that now what the immunized host can do is that it's got a whole army of T cells just waiting for that guy. And the thing is, because they don't have the neutralizing antibody anymore, that doesn't work. So the immunized people become infected. They don't really get very symptomatic because the interferon coming from the T cells is blocking the widespread replication of that virus in the body. So you don't have this cytokine storm and the whole inflammatory reaction gone haywire particularly in the lungs. And that accounts for the fact that the immunized people are much better off than the non-immunized people, which gives the raison d'etre to run out and get a vaccine to the ones that they've got now. And you know what's going to happen. They're going to get Omicron specific neutralizing antibody vaccines and so forth. But in the end, my prediction is that what we're going to need is that we're going to need a T-cell vaccine that will recognize gene products of the virus that are common to all the variants, because those genes and gene products in the virus are critical for the virus to stay alive and to replicate. And so they're going to be conserved, and they're not going to be, if they're mutated, those mutants just won't make it, and they won't be able to replicate, essentially. That's all a final way to say, send in the T cells. That's what you got to do. So I'll stop there, and thanks for listening to me. If you like this particular video, tell all your friends, number one, but number two, and send it to all your friends via electronically. But like it on the YouTube channel and subscribe to it so you'll get more, and click on the bell to get notified for more. And also scroll down on the YouTube site, look at my social media sites and so forth, and look at my website, where you'll find all of the classic papers in immunology on the writing tab, among other things that are there. So thanks so much for listening.